All right, today we're going to talk about cracking the code of life itself. I mean, literally, we're diving into the world of proteins. Those tiny, tiny machines doing basically everything inside our cells. But here's the kicker. How do they even get built in the first place? That's the puzzle we're going to piece together today.
So let me ask you this. If I just gave you a long list of parts, say for a super complicated engine, could you actually build the thing? With no instructions, no diagram, nothing? Probably not, right? Well, that is exactly the problem that nature solves, like millions of times a second inside you right now. That list of parts, that's just the protein sequence of amino acids. And the engine, that's the finished, working 3D protein.
And this really gets to the heart of it. On one side, you've got the blueprint. It's just a 1D string of stuff called amino acids, and our DNA dictates the order. But honestly, in this form, it's useless. It's just a floppy chain. It can't do a single thing. It only becomes the machine on the other side when it folds up into this super specific, really complex 3D shape. And that shape, that's everything. That's what lets it do its job. Getting from that simple blueprint to the working machine, honestly, it's like a magic trick happening inside our bodies all the time.
Okay, so that leads us right into our first big mystery. We're calling it the self-folding secret. I mean, how on earth does that blueprint, that simple string of parts, just know how to build itself, with nobody there to help it? How does that even work?
For decades, this was a massive puzzle for scientists, a real head-scratcher. But then a huge clue dropped, thanks to the Nobel Prize-winning work of a guy named Christian Anfinsen. He came up with this really elegant, simple experiment to ask a basic question. Can a protein find its own way home after you mess it up?
Okay, check this out, because the experiment was just brilliant in its simplicity. So first, he starts with a perfectly folded, working protein. Then he basically attacks it with some harsh chemicals. What this does is completely unravels it, right? Turns it from a working machine back into that useless floppy string. It's dead in the water, can't do its job. But then, and this is the incredible part, he just washed the chemicals away. And what happened? Bam! The protein just snapped right back into its original perfect shape, all on its own. It got back like 90% of its function. It was mind-blowing. It was like the protein remembered its shape.
So, what's the big takeaway here? It's absolutely crucial. Anfinsen's experiment proved one thing beyond a shadow of a doubt. The instructions are in the blueprint. That sequence of amino acids isn't just a shopping list of parts. No, it's the full-blown instruction manual. Everything the protein needs to know to build itself is right there in that 1D string.
But you know how science is, right? You solve one mystery and a new, even crazier one pops up. And that's exactly what happened here. This led to something called Leventhal's paradox. Suddenly, the question wasn't what tells the protein how to fold, it was how in the world does it do it so ridiculously fast? Right. So back in 1969, a scientist named Cyrus Leventhal decided to run the numbers. And what he found was staggering. He figured that for even a tiny protein, we're talking just 100 amino acids long, the number of possible ways it could fold is, well, it's that number right there. 3 to the power of 198. That number is so big, it's bigger than all the atoms in the entire known universe. It's just nonsensically huge. And that's the paradox. If a protein had to find its one correct shape by just trying out every single option one by one, even if it did it at lightning speed, it would take longer than the entire age of the universe just to fold one protein. But they don't. In reality, a protein folds in milliseconds, sometimes even microseconds. So, what gives? How are they solving this mathematically impossible problem in literally the blink of an eye?
Well, the answer is it's not searching at all, it's more like a guided collapse. The best way to think about it is this idea of a folding funnel. Imagine a ball at the top of a big, wide, bumpy funnel. It doesn't explore every single inch of the funnel's surface, right? No, it just rolls downhill. That's what a protein does. Physics and chemistry just kind of guide it rapidly down this energy landscape towards the very bottom, which is its one most stable, lowest energy shape. So it's not searching for the right answer, it's literally just falling into it.
All right, so we get the basic idea of how it happens, but how do we as scientists actually figure out what these crazy 3D shapes look like? Well, that gets us into some modern-day detective work. And there are basically two ways to crack the case: the old school way in the lab and the new school way with code.
And this lays out the two approaches perfectly. On one hand, you have the lab methods, things like X-ray crystallography. These are the gold standard for sure. They let you literally see the atoms, but man, are they slow and expensive. We're talking months, even years, and a lot of proteins are just too fussy. They refuse to be studied this way. Then you've got the other approach, computational modeling. Here, you just feed the computer the blueprint, that amino acid sequence, and it predicts the final 3D shape. It's way faster, way cheaper, and you can try it on any protein. The only catch is, it's a prediction. It's an incredibly educated guess, but it's still a guess, not a direct photo.
But here's the thing. In the last few years, these predictions have gotten so ridiculously good, they're absolutely revolutionizing biology. I mean, we are finally getting to see the structures of thousands upon thousands of proteins that were basically invisible to us before because they were impossible to solve in a lab. It's opening up whole new areas of research.
Which brings us to the bottom line, right? The big so what question. Who cares what the 3D shape of some tiny molecule is? Well, it turns out that knowing this, it changes everything, especially when it comes to medicine.
Yeah, let's just ask it straight up. Why are we pouring all this time, money, and brain power into figuring out these shapes? The answer is simple. A protein shape is directly connected to your health.
It all comes down to this. Function follows form. A lot of diseases, a lot of them, are caused by a protein that's either folded wrong or is just doing its job a little too well. So if you know the exact 3D shape of that troublemaker protein, you can do something amazing. You can design a drug that's like a custom-made key for a very specific lock, a molecule that will fit perfectly into a vital spot on that protein and just switch it off. That whole process is called structure-based drug design. And believe me, it is a total game changer for medicine.
And listen, this is not some far-off futuristic idea. This is happening right now. The drugs on this list are real, they are lifesavers, and they exist for one reason, because we knew the shape of the protein we needed to target. We're talking about everything from high blood pressure medication to the antiviral drugs that fight HIV and the flu. In every case, knowing the structure was the absolute key to finding a treatment. So, let's wrap it up here. Think about this. Thanks to things like the Human Genome Project, we have the blueprint, the 1D sequence, for pretty much every single protein in the human body. And now, with these insane new computational tools, we're figuring out what the 3D machines actually look like at an incredible pace. So the real question for the future isn't about what we know anymore. It's about what we're going to do with this amazing new power. What mysteries are we going to solve next? What diseases are finally going to be cured? The possibilities? They're just beginning.